Okay, great. Good morning. I'm Lydia Boudreau. I'm a Michigan Fellow and Assistant Professor here in the Ecology and Evolutionary Biology Department, and also a member of the Organizing Committee. And I'm delighted to have the opportunity to introduce our first early career speaker, Andy Rominger. Andy is currently a PhD candidate at Berkeley in the Environmental Science and Policy Management Department, where he works with Rosemary Gillespie, John Hart, and Charles Marshall. He earned his BS in Biological Sciences from Stanford, working with Elizabeth Hadley and Rudolfo Durzo. And for his honors thesis, entitled Both Neutral and Deterministic Processes Drive Community Structure, he won the Kennedy Prize for Outstanding Honors Thesis, given to only one thesis in the natural sciences. He has publications in a number of excellent journals, including Ecologia, Ecological Applications, Global Ecology and Biogeography, the American Naturalist, and Ecology Letters. And today he will be speaking about the statistical mechanics of biodiversity in evolving island communities. Please join me in welcoming Andy.
All right, is this on? Are we good? Okay, well thanks so much for having me here. It's really quite an honor to get invited to do this, and I want to thank the committee for doing that. So this is kind of a cool, lucky opportunity to talk after Rosie. Hopefully not the last time that gets to happen. So I'll just kind of dive right in. As one does, I'm going to start by talking about Darwin. And Darwin concluded on the origin with wondering, are there general laws that govern biodiversity? And he was talking about this entangled bank of vegetation, and after more than 150 years, we're kind of still looking. So this hasn't been unproductive. We've found some really cool mechanisms that are very important for the patterns and dynamics of the diversity of life, including assembly by speciation and extinction, kind of classic ecological timescale assembly. And this is all really relevant because we're changing our world fundamentally, and how these processes that limit biodiversity behave is really going to determine what our future world looks like. And we still don't have a firm grasp on what all these mechanisms are. So despite this kind of plethora of assembly routes and processes that are relevant, there's actually striking emergent similarity between lots of different systems. And kind of as a case in point, I'm going to go a little nuts and really quickly look at plants in San Francisco, planted plants, ornamentals, native predatory arthropods in Hawaii, and birds in the Midwest, and they all show this same pattern of the species abundance distribution. And this kind of emergent similarity across systems is not limited to just species abundance. Of course, it's species area, it's metabolic rate distributions, it's the association, the relationship between abundance and metabolic rate. So how can we possibly begin to predict what causes all this generality? And I think that, as biologists, a lot of us got into this because of the details of organisms and the way organisms interact with the environment, which is really a beautiful part about being an ecologist and evolutionary biologist. But to get at generality, I think we have to begin to really embrace abstraction, which is where ideas from statistical physics might begin to become handy. So that's what I'm going to launch into. And this is not an unprecedented idea. This actually dates back to very biological people. So Preston, who gave us the log normal, the log normal species abundance distribution, the way he was really thinking about it is that there's so many routes to getting an assemblage of species together in a community that if anything is going to be general about that, it can't rely on the details of any one system. It has to be really a statistical property of any system. And he got probably the mathematics a little wrong. It's probably this guy who got a closer handle on the mathematics. But the idea, I think, is really valid. And it begins to think about statistical constraint instead of constraints imposed by the biology of a system. And this is not your great grand advisor's t-test statistics, right? This is the statistics of the probabilistic properties of large aggregates of whatever you're looking at. So the way these probabilistic properties arise is you have some mechanisms that operate at a very micro scale. And from physics, you've probably seen stuff like this, but in biology, this is still really relevant. Organisms are machines for taking energy from the environment, fluxing it, and doing reproductive work, which we might as well call fitness. And through the process of all of these things doing this simple mechanism, and they've evolved, incredibly complicated and interesting ways of doing this over and over and over in different environments, you get a huge system that's fluxing energy, that's using it to make individuals. Those individuals have unique evolutionary relationships with each other. And in a very real way, the fundamental problem, the fundamental challenge of biodiversity theory is to figure out how energy is allocated to individuals and individuals to species. So we want to make inference about this, and going kind of with the legacy of Preston, we want to make inference about this in a way that's both naive and all-embracing of all these really valid mechanisms that we can document experimentally and in microcosms and mesocosms. So how do we do that? How do we make kind of mechanistically all-embracing predictions about this problem? There's a principle from information theory that we can use, which it takes known constraints on a system, and it uses these to then make as naive a prediction about the probability distributions that describe that system. So as a really simple example, if what we know about something that we're interested in is that it's bounded between two limits, the probability distribution we would infer is just a uniform. That's the most naive. It's making no predictions about disproportionate allocation of whatever this quantity is that we're measuring to one end of that spectrum or the other. So this mathematical framework was applied by Jaynes famously to derive most of classical statistical mechanics, and we can use it to derive a lot of what we might call macroecology, the description of biodiversity at a very coarse grain. So what we do is, I'm not really going to go into the details of it too much, except to say that the relevant state variables, based on this realization that the fundamental challenge in biodiversity is thinking about energy allocated to individuals, individuals to species, the quantities of energy, individuals, and species are state variables in the system. Those are what we assume is kind of unchanging. And then by applying the maximum entropy procedure to that set of constraints, we can derive probability distributions that give us relationships of species to area, of individuals across species, and of metabolic rates. So I'm not going to go into the math, but we do have an R package that'll let you interface with this theory at whatever level you want. It can be super high level, and you can just predict the distributions for your data, or you can get into the nitty-gritty of how this maximum entropy optimization works. Okay, so why would we bother to test theory? And this gets back a little to what Rosie was talking about. Brian McGill and Jeff Nekla had this cool paper that said mechanism just kind of seems to be AWOL in ecology. But what theory can do is it can give us a new perspective on old questions, on old data, and we can see whether or not those data are actually exceptional or expected. And that's exactly what this maximum entropy theory of ecology does. This is an example that shows how species area relationships, the slope of species area relationships across a huge number of different communities collapses onto one prediction. So it would imply that all these communities are kind of behaving as expected. They're behaving just like ideal gases behave. They're kind of at a steady state, constrained by the statistics rather than by the biology. But what's really cool is once we have a prediction for the expected, we can really begin to look at what are the unusual systems and what are the processes that drive those systems to be unusual. And the kinds of processes that I think we really care about, or at least that I really care about in biology, are the ones that impart memory, that impart historical contingency. They're the evolutionary processes that are so unique to biology and not seen in any other science. We don't have descent with modification. Like we don't have that in physics like we do in biology. So that brings me to using Hawaii to test these kinds of statistical equilibrial theories and look for deviations that are going to tell us cool and important things about the evolutionary process and how that drives community assembly. So you all are familiar with this system now, and just to recap, as these islands form, they get colonists, those colonists diversify, they diversify down the chain, colonization continues, This is all done in a trophic context with vegetation, that's the V, I kind of forgot what that stood for. Vegetation, detritivores, herbivores, and predators. And this has led to this spectacular radiation of diversity in this island system. So to capture that evolving ecology, I'm going to be using published data. It's about 16,000 arthropods that have been collected and identified. They were collected from canopy tree fogging, a massive project that samples across the chronosequence. And to start with, we'll just look at this abundance distribution. And I'm going to flip things around a little bit. We're used to looking at these rank plots. If you take the negative inverse of that, you get the cumulative density function from the underlying probability distribution. And so I'll compare this equilibrial theory based on maximum entropy to the real data. And to start with, we'll look at these detritivores across the chronosequence. And really strikingly already, you can see that the really young communities are the ones that deviate most from theory. And to put that in a little bit more quantitative terms, you can look at the mean squared error of theory versus data. And as you get younger and younger in the chronosequence, so younger is over on my side, kind of this is like the paleo axis switching. So young on the right, old on the left. As you get younger, you get more and more deviation from this equilibrial theory. But that's just for detritivores. We can look at the other trophic groups and they show disparate patterns. So that's really kind of cool. I'm going to calibrate the axes so the dashed line is what it looks like when it's really a pretty poor fit to theory. Solid line is when it's really a pretty good fit to theory. So basically, detritivores, the younger the community, the more the deviation from theory. Predators are well fit by theory across the board, and herbivores show the biggest deviation at the oldest sites, although they also deviate quite a lot at the young sites. So what could that be? This is where we really get into the nitty-gritty of the biological mechanisms. We don't have enough replication to look at this everywhere, but a really cool thing to look at is the beta diversity between samples within so between replicates within a site and also across sites. And that's what you're seeing here. These are normalized by null model simulations. So different trophic levels have different amounts of kind of intrinsic species turnover. And when you look at how that species turnover correlates with your deviation from theory, the more turnover there is, the more deviation from theory there is. And to me, that really implies that, what are the things that lead to higher turnover? It's lower dispersal, keeping in mind that these are all environmentally homogeneous sites. We selected them to have the same environment. So more turnover in space would imply more dispersal limitation, would imply that dispersal is really limiting to the assembly process. And that's probably what's driving the deviation from this equilibrial theory, at least at the young sites. And at the old sites, we don't know yet, but stay tuned because we're going to at least kind of tease at that a little bit. We can also look at the distribution of metabolic rates in the same way. And this is, again, with detritivores. And looking at that, I'm going to go ahead and calibrate the axes again. This is super qualitative. We have a new quantitative method of doing this that you'll see, but for now it's just qualitative. So dashed line is what a bad fit to theory looks like. Solid line is a good fit to theory. And again, now it's kind of cool. Detritivores show deviations from theory at old and young. Predators, again, are well fit all around. And I should note from population genetic data that even though Rosie showed a lot of differences in spider population genetics, when you compare it to these other trophic groups, they're much more dispersive than herbivores and detritivores are. So that really makes actually a lot of sense. But the cool thing to highlight here, this is how organisms are fluxing energy that they get from their environment. Remember, herbivores really stand out as deviating hugely from expectation, except for in this kind of middle-aged site. So what's going on there? Well, of course, the thing we know and love about herbivores is they have these really cool, potentially co-evolutionary or co-diversification interactions with their plants that they're feeding on. The plants don't want to be fed on, the herbivores want to feed on them. It must stay up all night worrying about that kind of stuff. But this is really cool work that's been done on herbivorous hemipterans. And around about 1 million years in the chronosequence, you start to see real niche exploration where these different clades are beginning to explore different host plants. And so we compiled plant herbivore networks from the literature on these herbivorous hermipterans. This is what they look like. It's pretty much completely meaningless, but it's pretty. When you actually compute with them, you can build another maximum entropy theory for networks. And what you see is that it's the, let's see. I don't want to push anything for, okay, that's it. At this middle-aged site, this density plot is the simulation of what you would expect the data to look like if the MaxEnt theory were true. And the red is what the data actually have. These are likelihood values, log likelihoods. So at this middle-aged site is the only place where the network of species interactions conforms to this statistical equilibrium. At all the other sites, it deviates quite a lot from it. So going back to the metabolic rate distributions, again, it's at the middle-aged sites where you get agreement with theory and metabolic rate. At the networks, it's middle-aged site where you get agreement between species interactions and the theory. If you do networks, you're probably more familiar with looking at nestedness and modularity. They have a cool contribution to this story as well, which is that at the younger sites, you have really high nestedness, and you can debate until the cows come home about what nestedness means. But one of the things that could be indicative of are these are dispersal assembled communities. So something enters into the system, it's figuring out who to interact with. It's going to more likely interact with a generalist than with a specialist, and that disproportionate interaction of specialists with generalists and generalists among each other is what nestedness is. So assembly by dispersal is consistent with this nestedness pattern. And then for modularity, it really picks up at these older sites. And modularity, there's some really nice publications showing both empirically and with simulation why that's likely to be the result of coevolution forming these kind of modules of highly co-evolved species. So there's kind of a complex of, in this case, herbivores that specialize on a complex of plants, and they're all kind of circling each other through this evolutionary process. So it's likely that it's, again, incomplete assembly. This is kind of maybe over-interpreting these nestedness patterns, but again, stay tuned. So incomplete assembly, again, driving this early deviation, and then the later deviations could be this super non-equilibrial evolutionary process of tracking your host and your host kind of running away from you through time. So to summarize all that, if you kind of squint your eyes and go to Mars and look at Earth from super far away, you can imagine community assembly looks like a linear stochastic process. Just imagine that. So if community assembly is kind of headed towards some stationary state, early on it's really limited by these rates, whatever they may be, rates of dispersal, rates of adaptation. And then later on, if you see deviations again from this steady state prediction, it's probably these really interesting non-equilibrial evolutionary interactions. One question would be, hypothetically, it's like a thought experiment. Could you assemble a community directly from the stationary state? And you could. What it would take is kind of going to the upper limits of dispersal and adaptation and somehow stripping away the evolutionary history of the community you're looking at. And how do you do that? I think you do that through biological invasion. So when we look at the proportion of the total metabolic energy in the system co-opted by invasive species, and we look again at the mean squared error of the theory, the more invaded you are, the more you conform to this statistical theory. So that's kind of cool. When we wipe away the history of the system, we get a better confirmation to the statistical steady state that we would expect. So how to actually get at that in more detail, it would be phenomenal to build theories that actually work directly with the best representation of evolutionary history that we can get with extant organisms, which would be a phylogeny. So I just want to mention, which Rosie brought up as well, that we're pioneering this approach we're we're modifying the pioneering work of other people. We're re-pioneering this approach to be able to take a huge sample. We've got on the order of 1 million specimens now to deal with, to take a huge sample of organisms, bulk extract their DNA, run it through your machine of choice, and then you would love, you'd love to be able to reconstruct the community from that. There's a really important intermediate step, which is dealing with all the biases that can arise through this process. So there's bias in the DNA content of different organisms, there's amplification bias, and in this project, we're actually building a reference library. So we're not going to use clustering algorithms or anything like that, but that's another source of potential bias. So you have to account for all that, and a really nice way to do it is given to us in the framework of hierarchical models, which are often implemented in a Bayesian framework for computational reasons more than anything else. So that's going to be something I try to tackle in my postdoc. And if you can really nail the statistical inference, you can reconstruct almost anything you want. You can reconstruct the phylogeny, you can reconstruct the food web if you are sequencing gut contents as well. And we're going to do that with these dimensions plots that it's been a labor of love to get everything sampled there. So we can look at a phylogenetic maximum entropy theory. We can look at phylogenetic patterns of species interactions, not only with the phylogeny, but with the phylogeny along snapshots in time. So I want to leave you with this question of what constrains biodiversity. And I think when the state variables are known, I think we actually have a fairly decent handle on this using this information theoretic approach that just says, if a system's in steady state, a lot of that biology gets kind of canceled out, and you're really just left with the statistical constraints on the system. Acknowledging that when those interactions aren't canceling out, when you're not in steady state for historical contingency reasons or for cool evolutionary interaction reasons, you get deviations that hopefully soon will be predictable. So in a way, we have a good handle on that, but what constrains those state variables? Why do we not have a world of microbes only that are super numerous and diverse or not, but super tiny, flexing tiny amounts of energy? And why don't we have this, Darwinian demon organism that's just everywhere and we have no diversity? To really get at that, I think we need a much more detailed interaction with these classical evolutionary ecological mechanisms. And it'd be awesome, maybe one day we'll actually have a statistical mechanics of diversification as well. Either you cringe or you think that's cool. But with that, I just want to say thanks again for having me here. Thanks to funders. Thanks to some really key collaborators on this. Be happy to take questions. Oh, no.
It's an easy question.
Okay.
So you said MaxEnt is good at predicting patterns of biodiversity, be it species area relationships or species rank abundance and stuff. What are the constraints that you need to put in the theory to get the right predictions that actually match observations? Is it constraints on average energy?
Yeah, it's metabolic energy, total number of individuals, and total number of species.
That's not a lot, right? It's kind of the type of constraint that you would think of putting in there, and then you get the expected answer. So the question is, if we can explain things that everybody is always asking about, like what explains biodiversity on my favorite system? And it happens to be, well, if you know the average energy, you can tell the number of species you expect there. Are we asking the wrong questions? Are we focusing on, like something that seems exceptional to us, but it's actually boring? It's like, that's what you would expect to see if there's nothing interesting going on in there.
I think in a way, yes. So two-part. One is that if it does fit with this really simple theory, it's certainly not that nothing is going on there, but in a real way, you don't have the information to distinguish between the relative roles of all these different mechanisms that you might care about leading to species abundance distributions or leading to metabolic rate distributions. It's not that those mechanisms aren't there, but you don't have the inferential power just by looking at these macroecological patterns. But the other thing is, if you do see a deviation from the theory, then go ahead, build a model that predicts species abundance distribution from your favorite mechanistic pool and see if it works. So, we're not the only ones saying that. You know, there's that paper in Ecology Letters with a huge author list that said, you know, we need to move beyond just species abundance distributions because there's not enough information in them to think about how we want to model community assembly. So you do need to bring in other data to distinguish between your hypotheses.
So you talked about deviation, one important source of deviations from the predictions as evolutionary contingency, historical. But of course, there are an awful lot of much shorter term causes for deviations. I mean, many ecologists think that the idea of systems being at steady state or, you know, an equilibrium community given constant variation, secular changes in climate, just all of these things is also not very reasonable. So is there an argument for kind of privileging evolutionary contingency over a kind of shorter term ecological contingency as an explanation for divergence?
Yeah, I think there's no, what was privilege? What was the word? Yeah, no, that's all good. There's no, yeah, what should I say? There's certainly lots of reasons why short-term stuff would matter. And in the lab, my good friend and lab mate Erica Newman's looking at how ecological timescale disturbance leads to deviations from the theory. So that's certainly very relevant. And again, you know, it's the same mechanisms. It's dispersal limitation and its species interactions, but they're playing out on ecological timescales. And yeah, the studies, I think I'll leave it at that. It's certainly relevant.
Adding just a little time depth to Deborah's question, since we're living in the Quaternary with an ice age and the climate's warming and cooling, then species distributions have been shifting a lot over the last 2.5 million years. Are you able to see signatures of, say, highly disturbed assemblages versus ones that have been potentially more stable over the last 2.5 million years in any of the analyses you've been doing, Hawaii or elsewhere?
I haven't done the analyses to look at that, but I think that would be a phenomenal thing to tackle. Yeah, really super good to do that. Maybe we can talk about what systems would be best to do that in. I will say part of what I'll get to in this postdoc coming up is using museum collection data to start to address that, because so much of the diversity at these deeper scales in time and broader spatial scales, you know, we don't have an ecological census for that. So to be able to build the statistical inference procedure to use those disparate or haphazardly collected data to make this do this kind of analysis would be great. So yeah, I think that's super awesome thing to look at in the near future. Cool. Yeah, well, we have a coffee break in a few minutes. So. All right. Thanks a bunch, folks. Appreciate it.